For the next few minutes, I'm going to talk about the genomics and bioinformatics in infectious disease. Though my title says that I'll be talking more about the AMR and how we are using AMR effectively to manage the patient. But it's in a small segment. I thought I will give you an overview in the last few years how the science data with the help of machine learning has improved or impacted, been very beneficial for the infectious disease control or intervention. So that's how I've slightly changed, and I'm trying to show you in the next few slides the volume of data which has been generated, and using the data, whatever the queries we raise to know from the data, machine learning or the various tools have been, applications have been developed, and it easily gets those information for us to improve the healthcare. So in the overview, how the lab diagnosis is improved with the health data and machine learning, the laboratory diagnosis, then antimicrobial resistance, and how we choose correct antibiotic to the patient, then how the infection control and prevention is done, especially using the surveillance data, and as well as outbreak investigation we achieve and intervene and reduce the infection spread. And lastly, most important application which I would like to mention here is its role in the vaccine design. So I would like to give few examples of the vaccine design which immensely helped to control the infections. So the role of genomic and bioinformatics in ID, one is for the diagnosis. It can easily identify the organism very early, because the conventional culture takes long time, up to 48 hours you have to wait, or even 72 hours you have to wait. TB you need to wait for 6 weeks. Whereas with the available molecular techniques and the genomic and bioinformatics all put together, within 1 hour you can identify the Mycobacterium tuberculosis positivity. If you wait for 6 weeks, then the disease would have infected another 10 to 20 people, and you have to wait for another 6 weeks for the antimicrobial resistance information to come. So it almost takes 12 weeks to institute appropriate therapy in those individuals. In 12 weeks' time, you would have infected many number of patients. So within 1 hour, we achieve identification of the mycobacterium as well as the drug-resistant profile of the mycobacterium, and we institute a proper therapy, appropriate therapy within the same day. So much of a transformation has occurred because of the genomic laboratory diagnosis with the help of machine learning tools. So this is the great revolution which has occurred in not only in microbiology, in cancer biology, and in radiology. These were all the 3 early areas where quickly we could come to diagnosis. So resistant virulence pathogen detection, and they have developed many number of kits. And pipelines have been developed. Next to the microbial diagnosis, optimizing the antimicrobial therapy. Because again, the antimicrobial resistance can be due to mutations or chromosomal mutations which can be identified using the genomic information, or it can be a plasmid, horizontal gene transfer, where you can again identify using the molecular methods. So then AMR surveillance, how the disease spreads in the hospital, in the community, or over the continents. So that can be easily identified. We all know that last 2 years we suffered with the SARS-CoV and various variants in each locality, how it came. Delta, China gave the virus, and we gave the Delta virus with high morbidity and mortality. Then South Africa gave the Omicron. So it's not that it occurred in those areas. So we could know in which region what was happening. So quickly we could get a vaccine done, and then from one monovalent vaccine we moved to the bivalent vaccine, and US FDA has approved that one. So this all was possible because of the health genomic data and machine learning tool which can analyze the data and give you the answer in real time. And then vaccine strategy, again what is the particular target for the vaccine. These are all the 4 important applications for the laboratory diagnosis, to choosing the right antibiotic, and to do the disease surveillance, and for vaccine strategy this was useful. Now I will little detail more on the laboratory diagnosis and management. Now you know that 3 different approaches are taken. One is from the small animals or the microbes, you do the whole genome sequencing and you have all the information about that particular organism. Most of the time, that is not your requirement. You would like to have not only one animal, you want to learn about all the entire zoo. That's the place where you do the metagenome. Whole genome, metagenome you do, or shotgun gene sequencing. But of late, what we are seeing is the more and more interest comes with the targeted metagenome. Here, what we do is, we don't want the entire zoo. We are interested in only certain animals, something like that. So what we do is, our interest, if suppose one organism is the SARS-CoV, we are interested in what lineage it is, and whether that lineage will be susceptible for the existing, it will respond to the existing vaccine or not. So only 4 or 5 points, important information from that organism we want. Then what we can is, we can tailor. That is called as deep amplicon sequencing or targeted metagenomics. So that's how we are trying to get the best out of both machine learning and the health information. So 2 classical examples which I would like to give is, the Oxford Nanopore developed a kit called the Midnight Kit, where within short period of time, you can do the library preparation, sequencing, and data analysis can be carried out within few hours. And this gives an consensus genome sequence for the identification of the variants. You know that you would have heard about the variants of concern, variants of interest, many classification WHO gave to follow up the SARS-CoV. So this is how the kit helped us to achieve so early what lineage it is and whether the vaccine is good enough or not and how to intervene in that one. So another TB is another important chronic infectious disease where drug resistant XDR TB is quite high. And from the sputum sample, 13 resistant markers can be identified within few hours. Otherwise, it takes 12 weeks. So this is achieved by, again, targeted whole genome sequencing. This is called Deplex Mycobacterium TB kits. So there are many number of kits and many number of areas are coming up. They are all more in the verge of getting FDA approved. So this is the revolutionizing happening in the diagnostic field. So now I move on to the treatment aspect, management aspect. India is the epicenter of antimicrobial resistance, and you know that every name the organism escape pathogens, which are all the hospital-acquired infections, E. coli, Klebsiella, Pseudomonas, Acinetobacter or MRSA, and TB. So these are all for Acinetobacter, XDR Acinetobacter. If you get that infection, 85% are drug resistant. And irrespective of the therapy, 40% will die. And this is all over the world. You name any country, this is the situation. And to identify early and intervene or to control the infection, you need to know the presence of that organism in your hospital environment. And you need to diagnose. So what they do is they do sequencing, and thousands and millions of organisms have been sequenced and a database has been created and it is shared, and they have developed a consortium for every organism, and people from every continent or every country are part of it, and with the data they give disseminating information, and new tools are developed to get whatever the information required from the data. And from the data they are developing a point of care test so that you can quickly recognize, identify and isolate the patient. And then for treatment aspects also, it gives you information whether monotherapy is good enough, or in the beginning itself you should go for a combination therapy. So these are all possible only with the data collection, genomic data collection, and quick analysis using the machine learning. And then personalized antibiotic therapy. So here what happens is, for any organism you have identified, we know whether it is an chromosomal mediated or plasmid mediated. Then easily you can identify the resistant bug, and knowing the resistant mechanism, then you know which antibiotic will work and which antibiotic may not work. For example, I have given you the carbapenem tailored therapy. If it is an OXA48, ceftazidime-avibactam alone is good enough. If along with the OXA48 NDM is there, then that is not sufficient. You need to add aztreonam. And in the next column, what I've given is, in addition to OXA48 and NDM, if you have a penicillin binding protein 3 insertion or AMPC, then even this combination won't work. Then you need to go for a phosphomycin combination. So to choose the right antibiotic, you need to know the genomic information of that organism, where in an automated platform, it will sequence and the machine learning will interpret and it will give you the result within 1.5 hours. That is the best component. It's available in affordable settings, and where it's the question of affordability and accessibility to the patient. We have all the right tools to identify the infecting organism and its resistant profile at the earliest now. So genomic AMR surveillance, this gives the information on empirical therapy. Empirical therapy is, you don't know what organism is infected, you guess and you give a broad coverage. If you know the organism, then you give the targeted therapy. So here you don't know what organism it is, so you need to start a broad coverage. If you use broad coverage, then you are misusing and overusing the antibiotic. As of now, there is no good antibiotic which knocks off all the resistant bug. So you need to judiciously use the empirical therapy. To choose the best empirical therapy, you need the genomic information. And infection control in the hospital. So now the hospitals are the big incubators. They are the one which causes high mortality and morbidity than the community infections. So you need to have a tight control of infection in the hospital, and sometimes outbreak either in the hospital or in the community it can occur, then you need to identify. So for that you need to have a genomic information, and to compare the genomic information you need to have a database, and this all can be quickly analyzed and interpreted by the machine learning. So in US they have a PulseNet program and the institution which takes care of this exclusively. And now not only within the humans, outside the humans, from the environment and from the animals, it's called One Health, there is a transmission of the infectious disease. SARS-CoV is one. From the bat and the pangolin combination, it came to the human. So it's a cross species. So something like that, that's why One Health is given more importance. So we identify all the resistant mechanism from the whole genome sequencing, and we do interpret. And how do we control the infection? So once we do the surveillance, we do the culture and susceptibility, and we monitor the trend of the susceptibility, and we look out for any emerging resistance, and we look at the population structure and look for the evolution and the dissemination of the resistant lineage. So if it is spreading, then it's very important for the hospital and the county to stop the dissemination or the spread of the disease. When we are doing it in the hospital settings, simultaneously it needs to be done at the outside the hospital. Mainly the major problem now we are facing is from the poultry. That's where the poultry are fed with a good volume of antibiotics, and multi-drug resistant organisms emerges from there. And that's another breeding center. And the role of genomics and bioinformatics for aptness of the vaccine. So I have given 5 different vaccines here, 4+1 SARS I have missed out here. And I will tell you how it has improved the quality of the vaccine or the efficacy of the vaccine for the betterment of the existing old ones. So first example I would like to give on Salmonella typhi, and there was extremely drug resistant Salmonella typhi, where even it was resistant to 3rd generation cephalosporin. And this occurred in Karachi, Sindh province of Pakistan. And luckily by then, within 3-4 months, all the international agency converged to those places, collected the sample, did the whole genome sequencing, and they identified that it belongs to certain lineage, H58, and they know the resistant mechanism has been identified or deciphered, and by then the vaccine also was readily available. So preemptive means infected area, the geographical region and the surrounding region were preemptively vaccinated, and they could control the spread of extremely drug resistant Salmonella typhi. And this was a great first time success where they could contain within a country for an infectious disease. And then further it was taken up in certain geographical regions selectively, the hotspots in Liberia, Zimbabwe, and Navi Mumbai, where the XDR-S typhi has been very impressively controlled, and no more we are seeing. Very little spillover is there, but still it was one of the best example where effectively it was controlled. And another important vaccine is Streptococcus pneumonia, which causes pneumonia, which is called as captain of all death. So especially in the younger age group and in the elderly age group, to prevent infection, what we do is we give various valent vaccines. Pneumococci has got 100 serotypes. In the 100 serotypes, first for nearly 60 years, because genetics was not there and machine learning was not there, and they were not knowing that it has many number of serotypes. They thought it's only one serotype, and 60, 70 years of research failed. They could not develop a vaccine. The moment they came to know that it has gotten a different serotypes, my professor said that when he started his MBBS, it was 4 serotype, and when he post-graduation, it has become 20. When he became professor, it has become 67. So similarly, when I knew it was 80, and now it is 102. So over the time, the number of variants, like SARS-CoV variants, the serotype, now it's 100. And the vaccine available is 7-valent vaccine, 10-valent vaccine, 13-valent vaccine. Now 15, 20, 25 are there. So which one wear what they want? You need to have a surveillance data, genomic data. If you have the genomic data to that particular locality, you can choose which vaccine is required for that locality. So what works better, 10-valent works much better in the US, but it doesn't work. Under 90% effectiveness is there, whereas in India it has got only 60%. Though you need to add a few more serotypes to that vaccine. So that's how you need to move to the another high-valent vaccine. So how this happens? Because the AMR and the capsular gene are very close to the gene, and a lot of recombination occurs. One example I can give you is among the 100, 10D is the new 101 serotype. When you decipher the detail of that structure, you can see it has picked up from the serotype 39 and the serotype 8C and formed a new 10D. So this could be possible only with the genomic data and interference with the available data. So again, what the organisms keeps evolving. Sometimes they lose capsule, sometimes they inherit the AMR, then they get the capsule, then they keep changing. So serotypes also keeps evolving. So variants of each organism keeps rising. So it's a continuous evolving bacteria and human. We need to keep following up, and according to the evolution, we need to match the treatment or prevention with vaccines. So now diphtheria, for the past 50 years, we are using the vaccine made up of from the single strain, same single strain we use for manufacturing the vaccine. But the circulating diphtheria in the community has changed. So it has mutated for the toxin gene. So it may not work for that particular vaccine. So vaccine, in spite of taking vaccine, there will be a vaccine failure. So that needs to be identified. To identify that, you need to have data. From thousands of people, you should have collected the isolate, should have done the whole genome sequencing, and you should look for the mutation. These are all manually, it's not possible. It will be possible only through genomics machine learning tools where you can pinpoint the problem. So that's how for diphtheria and pertussis, we have identified the mutation, and the mutation has to be validated that it is significant and functionally it's the one which has caused for the failure. So we identified, and once we know that it's failure, and we will overcome that mutation with either fixing up something, engineering that bacteria, or we will go for another new strain. And now we are very close to eradicating the polio, but still we have not eradicated the polio. Now all the polio cases, polio paralysis is due to vaccine strains, vaccine-derived polio. The strain which is used for oral polio is causing it's a very low in number. So its benefits are much higher compared to the ill effects. Nearly last year, 1,000 cases were identified. Polio cases were identified across the world, and almost all the 1,000 are due to vaccine strains. So how to get rid of that one? Best is Salk vaccine, but it's not cheaper and it's not easy to have. Whereas the oral polio is the cheap and easier to give. So Bill Gates comes in, and 20 years of science helped us to identify the mutation, and they have engineered the virus to fix up that mutation. They modified the 5-UTR region, they stabilized the 2,6 coding region and 3D polymerase, and the new engineered strain was developed, and then that was put into animal model, tested for its safety, immunogenicity, then given in the human. Again, the safety and immunogenicity was successful, satisfactory. Now the engineered strain is used to manufacture the vaccine. Now that vaccine is being used in 27 countries. That's how it's evolving. So all this large-scale global impact is happening only because of the genomic data generated across the world, and where people have access to it, interpret that information using the machine learning, and they could arrive at fixing up this problem as a solution. Similarly, SARS-CoV, I told you earlier, first we were with the Delta wild type, and we have the quick vaccine for it. Then Omicron came, then quickly the vaccine manufacturer went for an additional antigen into it. Now it has become a monovalent, has become a bivalent. So this is how it keeps evolving. These are all possible only with the huge health data and machine learning. As of now, we are not seeing much of an artificial intelligence coming into the science, because there are certain concerns which I will address you later. But as of now, the machine learning has really, it's a game changer in the medical science. And a few more I would like to add into the list. One is how we take the antimicrobial susceptibility testing. It keeps reading the zone of inhibition many number of times. It keeps the memory, and quickly within few minutes it gives the interpretation. Not only the moment it reads, it also can identify the resistant mechanism and give the minimum inhibitory concentration, so that you can choose the right antibiotic at right dose and you can give for right number of days. The absolute antimicrobial stewardship can be implemented with great ease. And there are many organisms where from the animal species will cross over to the humans. So when it comes, initially it may be localized and subsequently get adapted to the human, and it becomes invasive. So how to predict that? Again, the genomic information gives something called the invasive index calculator. With that, we can be to great extent predict that this will be a pathogen in a near future. And the drugs can be identified. Suppose if the drugs are not working due to some mutations, then we can be sure that that mutation is significant, and to overcome that mutation, either new drug or combination drug can be given. So that's how for breakthrough drug discovery, again genomic data and machine learning greatly helps. Now people are thinking that machine learning coupled with artificial intelligence, whether that can solve the problem and you no need to consult a doctor. But of course, that's not possible at this stage. And the concerns are, I will just list it here. So now to conclude this talk, definitely genomic and the bioinformatics aids in the laboratory diagnosis within few hours. It doesn't take days to weeks, number one. And it helps us to give the start appropriate therapy, thereby reducing the mortality and morbidity. And it helps to the large scale surveillance. It reduces the infection spread. And any outbreak investigation, it helps to identify the source of the infection. And many other potential applications are there with the data generated. What is the role of machine learning and artificial intelligence at present in infectious diseases? Most of the machine learning applications are very useful and it's in great use, but artificial intelligence's inputs are very minimal as of now. Second one is the machine learning success has come mainly because of the huge volume of data generated and how you teach the machine learning. If you have taught or trained the machine much better, then the benefits you reap are very high. So two key important features which we see is the data generated, the volume of the data, the bigger the better, and the training of the machine. More inputs you have given, taught well, and you have educated well, then it interprets, gives you the output much better. So that is the trade-off now, where you invest, you will reap. So that's the one. And the artificial intelligence, as of now, they have tried many things. Biggest alarm what they have given is patient safety concern. So even 1 in 1,000 goes, mistake happens, then it's not acceptable. So as of now, it's evolving science. Once we achieve better and over the time, if certain places, definitely artificial intelligence will be helping us, but not in all the situations. That's what even earlier Dr. Menon was mentioning about that. With that, what I would like to conclude is, now for us the clinician, this language was like Latin and Greek earlier. Now for a clinician coming to the lab, bench work, then molecular work, then moving from the molecular work to bioinformatics, now artificial intelligence, the transition was quite tight. But still we have evolved, and this has definitely benefited the improvement of the healthcare. With that, I would like to conclude. Thank you.
Thank you, Dr. Balaji, for an informative talk on machine learning and its role in infectious diseases. Since we are running short of time, probably we'll take only one question from the audience.
Just one quick question, Dr. Balaji. This has been possible here. What is the arrangement in the country? You showed some, there's a map of India and there were certain states or centers which were delineated. Is the service available across? Is there a national program?
Yes, there is a national program by the Indian Council of Medical Research. They have given 4 centers they have chosen. Jigmar Pondicherry, CMC Vellore, then Chandigarh, PGI Chandigarh and All India Institute of Medical Science. We got a couple of centers and we generate the data, and since we are leading in the genomic front, most of the data are with us and we do the interpretation and we give them the national guidance for appropriate therapy. And in turn what we do is to improve our own knowledge, skill, and to get acquainted with the latest tools. We network with there are separate identities are there for every organism. It's just something like a super specialty. For Salmonella typhi, Shigella, it's the Cambridge Sanger Institutes are taking care. And for escape pathogens, the Monash University have their own database. And for Pseudomonas acinetobacter, they are the consortium head leading. So whoever is specialized in whichever the area, we network, collaborate with them, and we are part of the team. And we get their inputs and we also share our data with them. So now we have a good understanding of how common disease pattern is between countries and us, and how we are different, where the Western literature teaching may not help us, where we need to learn from our own, and we have to design our own therapy or strategy. We are very clear about that as of now. So really, hugely it benefits. Thank you.